Differences, functions, structure, and properties of DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Nucleoside, nucleotide, and nucleic acid in terms of structure and function. Nucleoside. Nucleosides are the structural subunits of nucleic acids such as DNA and RNA, composed of a nucleobase or a purine and a 5-carbon sugar. It plays an essential role in intermediary metabolism and biosynthesis of macromolecules. Nucleosides are responsible for encoding, transmitting, and expressing genetic information in all living things. Nucleotides. Nucleotides consist of a nitrogenous base, a 5-carbon sugar, and a phosphate group. Nucleoside triphosphates play a central role in cell metabolism. They are constituents of DNA and RNA which store and transmit genetic information. Nucleotides are also important coenzyme catalysts. Nucleic acids. Nucleic acids are the most important macromolecule which stores genetic information in biological systems. They are responsible for encoding all the virus-specific macromolecules of the pathogen. There are two main types of nucleic acid, which are DNA and RNA. These store the information needed by cells to create proteins. Structure and properties of purines and pyrimidines. Purines and pyrimidines are the nitrogenous bases which make up nucleic acids, the building blocks of DNA and RNA. There are two main types of purine, adenine and guanine, which occurs in both DNA and RNA. There are three main types of pyrimidines, but only cytosine exists in both DNA and RNA. The other two are uracil, which is the RNA exclusive, and thymine, which is DNA exclusive. The number of rings in the base determines whether it is a purine or a pyrimidine. Purines have a double carbon nitrogen ring with 4 nitrogen atoms, while pyrimidines have a single carbon nitrogen ring with 2 nitrogen atoms. The most important function of purines is base pairing. They form hydrogen bonds with the corresponding pyrimidines on the opposite strand of DNA and vice versa, holding both DNA strands together. They pair together through complementary pairing based on Chargaff's rule, which means that whatever purines pairs with whatever pyrimidines is always constant, as is the number of hydrogen bonds between them. For instance, adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds. The major differences between DNA and RNA. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. DNA allows for long-term storage and transmission of genetic information, while RNA is used to transfer the genetic code from the nucleus to the ribosomes to make proteins. DNA has a B-form double helix. It is a double-stranded molecule consisting of a long chain of nucleotides. RNA has an A-form helix. It is a single-strand helix consisting of shorter chains of nucleotides. DNA has a deoxyribose sugar phosphate backbone, while RNA has a ribose sugar phosphate backbone. DNA is self-replicating, and RNA is synthesized from DNA on an as-needed basis. The CH bonds in DNA make it fairly stable, plus the body destroys enzymes that would attack DNA. The OH bond in the ribose of RNA makes the molecule more reactive. It is not stable under alkaline conditions, and the large grooves in the molecule make it susceptible to enzyme attack. DNA is susceptible to UV damage, while RNA is resistant to UV damage. Transcription and translation. Transcription is the process where the genetic information on a DNA strand is transferred into an RNA strand by a series of polymerization reactions catalyzed by enzymes called DNA-dependent RNA polymerase. Translation is the process of protein synthesis where the information on RNA is expressed in the form of polypeptide chains. Transcription is the first step in gene expression, while translation is the second and final step of gene expression. The raw materials of transcription are the 4 base pairs of RNA: adenine, guanine, uracil, and cytosine. The 20 amino acids are the raw materials of translation. Promoted DNA sequences initiates transcription that signifies the beginning of the gene, while in translation, the binding of mRNA initiates the translation to the ribosomes. Elongation of RNA sequences occurs by the binding of complementary base pairs to the new sequence in transcription, while in translation, the elongation of protein occurs by the binding of amino acids. The product of transcription is the mRNA molecule, which is complementary to the DNA strand. And the product of translation is the peptide sequences encoded from the mRNA sequence. Transcription results in the synthesis of RNA sequences and occurs in the nucleus of eukaryotes and in the cytoplasm of prokaryotes. Translation results in the synthesis of proteins and occurs in the cytoplasm of prokaryotes and in the ribosomes on the endoplasmic reticulum in eukaryotes. This is the end of the presentation. I thank you for listening, and these are my references.